Thanks, Angelica. I want to thank the organizing committee, ESMTB conference organizing committee, for inviting me to give this colloquium. As Angelica said, yeah, it's based primarily on my research on reaction diffusion systems, and I want to bring to your attention the role of domain growth and cross diffusion. There have been quite a lot of, for many years now, quite a lot of criticism for Turing's theory and its limitations, and I will try to give you some sort of information or results on how we can relax some of those conditions to allow for general patterning. Yeah. So the take-home message at the end of this talk is, given a classical theory for pattern formation, it's well known that in the literature you have an activator-inhibitor mechanism. That's what you need. Should be long-range activation, short-range inhibition, where the diffusion of the activator is much, much larger than the inhibitor. And therefore, you have this long-range activation, short-range inhibition. The idea now in this talk is to illustrate that we can overcome this mechanism, and therefore, due to domain growth and cross diffusion, we can have what I call non-standard mechanisms for pattern formation. In the case of short-range activation, long-range inhibition, activator mechanism, for example. I'll give you an example of this mechanism as an illustration of the role of domain growth. Patterns are only forming due to domain growth. In the absence of domain growth, nothing, you will not get patterns at all. I will then also speak about inhibitor mechanisms. You can have patterns as well. If you have cross diffusion, you can have patterns with equal diffusion rates. And that's whether it's activator inhibitor or activator inhibitor, you have patterning, which is not possible on stationary domains. Yeah. So here is some of you might not know exactly what I do every day, but this is a slide just to say what my research group focuses on. So I'm interested in data-driven modeling. So from experiments all the way to validation, back again in experiments, where mathematics play a crucial role in that transition. So I'm looking at data-driven coupled back surface extracellular metric models for cell motility and pattern formation. And the aim is to develop mathematical and computational models for cell motility and pattern formation. And I want to couple two things, which are mechanics and biochemistry. So what is the area I'm looking at that allows me to do this? It's cell migration. So this is, again, a schematic diagram of a cell. This is a 2D representation. Think of this as a cross-section in a uniform environment, which is omega E here. It's a time dependent because the cell is moving on this environment. Sometimes if it is complete, if it is a biological environment, then it has to deform, the cell has to deform this in 3D in particular. And so everything here is time dependent in terms of my domain. So the interior of the cell I denoted as omega I of t. I'm completely ignoring a lot of things here about cell, the cell structure, cytoskeleton, and so on, intermediate filament, the nucleus. There's a whole lot of other processes happening inside. I'm just going to be very crude, and I consider my domain to be just uniform domain omega I of t, and it's evolving. It's enclosed by a closed boundary omega t, gamma t, sorry, and this gamma t it's a closed manifold really. Either in two, it's a curve in 2D and it's a surface in 3D. So think of a ball that's you can change in 3D, but these are cells really. This schematic representation is very, as I said, it's just a representation. So I want to look at mechanics that describe the interior of the cell as well as the exterior, the environment. And typical models are viscoelastic, poroelastic, and so on, depending on what you want to solve. Either you want to look at just the membrane or the bulk, which is the interior of the cell, the cytosol, and so on. So once I have that, in many cases, the mechanical processes are driven by the biochemistry for polarization, receptor ligament processes. And in particular, I'm interested in the dynamics, spatial temporal dynamics of raw GTPSs. So these are, in some of my studies, I've linked this with the mechanics. I could give another talk on that, but this is again a representation of some of the work that I'm doing. Yeah. So once we have the models, we derive these models, as I said, from first principles. We have the models, you have to carry out mathematical analysis where you can. And in many cases, we cannot get analytical sort of solutions, and therefore we resort to numerical analysis. And developing accurate, robust, and efficient numerical methods is very important. And not only that, also the methods have to be solved efficiently if you want to be able to fit the models to data in realistic timescales. Once you have the models, I should not use this mouse. Once you have the models, then you can look at validation. As I said again, I'm really interested in working with experimental observations, and I work with experimentalists. I will need to validate my models back into experiments. The other thing that's really important is these models have parameters, many parameters, and identifying values of these parameters is a challenge. And in many cases, in some cases, I want to sort of infer these parameters if I have data, given a set of number of models that I can develop for a specific observation. And then you can use Bayesian or optimal control approaches to infer, to estimate parameters by fitting the models to data in an optimal sense. Yeah. So that's really my work. And the talk today, this is the contents, but I'm not going to go through all of it in terms of what I'll cover, but I will cover analysis. I will illustrate with numerical methods using finite elements in this case, the theoretical findings, and then I'll conclude. Yeah. Here's an example, I think, of the role of domain growth in pattern formation. This is a picture from a Nature paper by Asai and Kondo in 1995, where they looked at a juvenile fish, a Pomacanthus fish, which has 3 stripes at a juvenile age. When the fish doubles almost in size, you have circular stripes emerging as the fish is growing. And as it grows to adulthood, you have more sort of circular stripes emerging. It's important to observe that the circular patterns also follow the geometry, so the curvature is important, geometry is important, doubling of stripes is also important. And so it's a clear illustration that domain growth is important in terms of the formation of patterns. I have here a movie again from Kondo, again a Pomacanthus fish. If you live, let's assume we leave this in a tank and we go on holiday. We're almost summertime now for all of us. We might go on holidays, maybe 1 month, 2 months. So you have bought this fish, you leave it in a tank, and hopefully you ask your neighbor to look after it. Yeah, he can put food maybe every day or something like that, feed it, and yeah, you take 6 months off. On a sabbatical, we go on sabbaticals, and you come back and you open a door, and in the tank, this is the fish that you find. And probably the first thing you will say is, okay, I have to ask the neighbor, what happened? Did he exchange the fish? What happened to the fish? Luckily, we live in a real-time world where we can video record some of these processes. And so by chance, you had your recording going on behind, and you switch it on, and you observe a patterning in real time from circular stripes to stripes as the fish is growing in real time. So this is quite a remarkable evolution of pattern formation during real growth development. And so I find this very exciting. But there are lots of questions here that I cannot go into during this talk. But this is an illustration of the importance of domain growth. It's a little bit difficult to illustrate the effects, the impact of cross-diffusion. I don't have an example in that sense. I'll try to find one if I can, but it's not easy. Again, Kondo shared with me these results in 2007 when I was looking at developing some of these theoretical frameworks. So you have an eel fish here, so you can measure and you can see a single spot here in the first image, which splits into 2 spots, and as the fish grows, it bifurcates again into 4 spots. If you have seen the work by Edmund Crampin from 1991, 1999, and also 2002, he published a very remarkable work on period spot doubling. He called it spot doubling during domain growth, and this was illustrated in 1D as well as in 2D. But when all this was published, there was no evidence whatsoever of being, can you observe this happening in nature? And Kondo illustrated this in 2007. So there's a need really to understand pattern formation during growth development. So let's go to the model. What is the model that I'm looking at? I'm going to assume that in the absence of cross-diffusion in the first place, that I have 2 molecular species, U and V. They react and diffuse through this semi-linear parabolic PD. It's a reaction-diffusion system, which I have non-dimensionalized. So I have the rate of change of U and V on the extreme left. I have terms that emerge due to growth. You can derive this using transport theorem on growing domains, on time-dependent domains, and V is the material velocity here. I'll talk more about this, and in particular, when I work with experiments, this material velocity V comes from experiments. You derive it from experiments. Yeah, but for the purposes of this talk, it's going to be a theoretical consideration of this material velocity. And then I have the diffusion. Yeah, as I said, I have non-dimensionalized. So D is the ratio between dV and du. In particular, V, I consider this as an inhibitor, and U is the activator. And again, I should intimate that this D generally understand that Turing theory should be bigger than 1, should be positive, also bigger than 1. Gamma is a scaling parameter that's associated to the length scale. F and G are reaction terms. That's where U and V only react and interrupt. Everywhere else, they don't, yeah, unless you introduce cross-diffusion. And I will illustrate that in the end. So I have a system of 2 PDs on a time-dependent domain. Yeah. The question now is, can we understand how we want to understand the solution behavior of this system? This is on a time-dependent domain, but it's something that's new. We don't know much about it in terms of the theory. So the idea in the following exposition, I will try to illustrate how we transform this PD on a time-dependent domain to a PD on a stationary domain, in particular the initial domain, such that my PD has time-dependent coefficients, resulting in non-autonomous systems of reaction-diffusion systems. Yeah. So I'm going to pay the price of, rather than working on a time-dependent domain, I'm going to work on a stationary domain with time, where my model, my reaction-diffusion system, has time-dependent coefficients. For simplicity, I'm just going to write it in vector metric form of this, in a compact form of this nature, and the notation is given here. And then it's easier for the analysis. I'm going to now map, as I said, this is the first assumption. I'm going to map my PD onto a time-independent domain. I call it omega naught. Yeah. And the assumptions are this psi, which is related to x through these bijective mappings. Yeah, mapping. So a standard choice, and this is really the assumption that I'm making here. It's called assumption 1 here. I'm going to assume that the rate of change of x, you can think of this if you're a computational biologist as your mesh. The rate of change of my domain is given by the flow velocity. But this is really a simplification, simply because we want to do some analysis. If it is not, and I put a note here, then you might want to think of arbitrary Lagrangian Eulerian framework. It's an open problem. I haven't done this, but I think this can be done where the rate of change of your domain is not exactly identical to your flow velocity. Yeah, so that's an open problem, one that I wish to highlight. Then the next thing, which is assumption 2, is to assume that my domain, I have a time-dependent domain that is growing or shrinking uniformly, isotropically, in a linear fashion. I'm going to ignore all other terms, rotation of the domain or translation of the domain. I'm just going to assume the simplest form, in that my flow velocity is related to, think of a balloon, you're blowing the balloon with air, and the rate at which you're blowing that, it's S of t, and the x is, you're changing the x as you evolve. Yeah. So if I do that, I can map now my PD. I have given you to a PD now posed on omega naught with time-dependent coefficients, h of t here and phi of t. The rest of them are as before. The parameters gamma, you don't see the d because it's embedded in this diffusion coefficient d here. It's a metric now. Yeah. So I have this vector metric equation. In particular, this is the d times, this is the Laplace operator really. Yeah. What is h of t is normal than the divergence of the flow velocity. So this represents either contraction or expansion of the domain. I've assumed uniform isotropic. I'll give you an examples of what these functions are for linear, for example, exponential growth, and for logistic growth. So I can compute this explicitly. Q of t is related also to the domain, and I can define it in this manner. Yeah. So that is related to the rate of domain growth really. You can see S of t is here and S of q are integrating really. Okay. So what are open problems in this area? It's nonlinear growth profiles. I haven't looked at the analysis of nonlinear. If you have nonlinear evolution of your domain, there's no analysis at the moment. We have solved computationally with Barrera and Elliott, where we looked at evolving biological systems, where our evolution law was driven by the molecular species as well as curvature. But we don't have an analysis, any analytical results for that. So it's something that I would like to do given time. There's no time, but you can only do so much. So so yeah, so that's it really. So before, what else do I need? Before I go any further, I want to identify, and this is important, I can identify a small order parameter epsilon, which is the ratio of the timescales of the dynamics versus the timescales of the growth, and I can show that this is small. This will allow me to do asymptotic expansions and to be able to linearize, sort of get asymptotic solutions somehow, close to bifurcation points. And so that's important. What else? Once I have this, and then I need to give you some definitions of what, this is the theory is similar to what you observe on stationary domains, but there are some caveats here. To start with, we are not talking about uniform. When we talk about critical points, they are not stationary points as fixed points, but we are talking about manifolds, time-dependent manifolds, which are our steady states. They are not really steady states. It's a time-dependent, spatially independent solution, US of t, which satisfies this system of ordinary differential equation. In the absence of domain growth, h of t will be 0, u of s, if it is a uniform steady state, this derivative will be also 0, and therefore you are only equating your kinetics to 0. And this is what we do when we compute uniform steady states. We equate our reaction terms to 0, and we find the uniform steady state. But this is not true here. We have to solve a system of ordinary differential equations in order to find your time-dependent manifolds on which you are going to carry your linear stability analysis. Yeah. So the system will either equilibrate to this or diverge from this. Under appropriate conditions, it's going to converge to a spatially inhomogeneous solution, which is the Turin solution that or pattern that you're looking for. And this is the definition that I give here of diffusion-driven instability on growing domains, which occurs when a spatially independent vector solution, US of t, which is linearly asymptotically stable. In the absence of diffusion, it becomes unstable when you add diffusion. Yeah. So that's basically it. So I can map now using linear stability analysis, I can map from a nonlinear PDE to a linear PDE. And this is the PDE that's given in W here. Yeah. And the question now is, the Laplace operator, I use the classical transport operator approximation with eigenfunctions and K squared are the wave numbers. So I can plug that. And now I will only have a system of differential equations. Now, what is W? I will decompose W in power series and therefore have a system only of ordinary differential equations. Non-autonomous, but it's a system of ordinary differential equations. And that's what I've done here. Yeah, I will end up with this system A, this is A dot of K, and it's a system of just ordinary differential equations. Yeah, you have your wave numbers here, you have your eigenfunctions, phi psi of K, and your coefficients AK of T, they are time dependent. In order to get rid of this H of T, which I have here, I can decompose my AK of T modes into BK of T times QK. And I plug it back, differentiate, and I can realize that this is going to be true. As long as Q of T is associated with the growth function, the domain growth, yeah, through H of T. So once I do that, I'm going to continue again. I can write this nicely, or I can write this nicely in terms of just vector metric notation again, as dpk of dt times mk of t times bk. Mk of t is defined here. So it's the metric that combines the diffusion part and the reaction part, the linearized evaluated at the time-dependent manifold. And if you look for solutions, we always do this on stationary domains or not. We always look for exponential solutions. Bk of t is an exponential solutions with lambda k, which are the eigenvalues, yeah, times some ck of t. If these eigenvalues, lambda star k, they are eigenvalues of largest eigenvalue, of course, they are time dependent of mk of t. If all of them, for all time, if they are negative, then bk of t will tend to 0, and this w of k of t will tend to 0, and your solutions, your u, will tend to us of t. So us of t will become asymptotically stable under some conditions. Now, we have ck of t here. It's important that we want ck of t to be well behaved, otherwise these solutions don't exist. So ck of t must be bounded. It should also be bounded from below by 0. It should not decay to 0. And I'll illustrate that. What else shall I say? In order to get patterning, we need this lambda k, the real parts of this, to be positive for some k wave numbers. Yeah, we need this to be positive in order to get exponential growth that will give rise to pattern. Okay, so given that I'm already running out of time, I will not go through the analysis, but we can show, and this is important, I'll show you just on the fly, we can demonstrate that there is exponential growth when the idea is we take a small time into the future. We freeze the system and we apply the asymptotic expansions, and we demonstrate using Picard iterations. You can cook up these solutions, you can construct them using Picard iteration, and you can demonstrate that the solutions, the eigenvalues, they are the leading orders, they are positive. So for some k for some wave numbers, and therefore you have exponential growth. I will also demonstrate again on the fly that the ck of t, they are well behaved, they are bounded, and they don't go to 0. They do not decay to 0. Yeah. So, but this depends on the eigenvalues. So let's go to this. In order to do all this analysis, you need two things. You need to define some measures. Of course, I have defined here ck of t given by this measure. I have also decomposed my ck of t in terms of two eigenvalues. One is a 0 and the other one is a negative, and so on. I can define a metric norm, which we know quite, it's pretty standard. It allows us to define the Lodzinski's measure, which is given here. And this, with all this, I can now demonstrate that the rate of change of this ck of t, of course, I'm going to use Cauchy-Schwarz inequality here, is bounded by the initial data. And this is important. The ck of t, the norm, is bounded by the initial data. So if you choose your initial data to be well-behaved, then CK of t is well-behaved, it's bounded. So that's the first one. I'm not going to go into the details of all this. At leading orders, you can show, and this is important, I've done it here, I'm using Picard iteration, you can show that CK of t is not decaying, is not going to decay to zero. These terms are, the leading terms are non-zero. Yeah, so that's very good. Similarly, you can demonstrate that in your the integral of these lambdas, you have growth, at leading terms of delta t. Really, you have positive quantities, and I define them here as the growth rate at the bottom, which is lambda star of t naught plus delta t of 2, and therefore with that you can compute the norm of the solution as when you take a small time into the future. This BKT solutions, you can show that they have exponential growth, which allows you to move away from the US of t, which is the time-dependent manifold, and your system is going to move to spatially inhomogeneous solutions. Yeah, but this is only for I've done this for when mk of t is real and distinct eigenvalues. You can do the same thing for complex, for repeated roots, whether real or complex, and so on. But I'm going to skip all this because there's quite a lot of information here. I'm going to skip it. But if you want any results, I can give you. Similarly for repeated. So in order to state the theorem that I want, the key theorem, I'm just going to take to denote T1 to be half of the time step when I move a delta T time into the future. Small time step, really. And therefore, I can define my H. H at T1, remember this is H is the divergence of the flow velocity, so I can evaluate it at T1 and I can denote this by H star, but it again depends on whether MK is real or complex eigenvalues and so on. Yeah, so I can compute, I'll show you what these values are in the end for some examples. So this is the key theorem really for my talk. It's a Turing diffusively driven instability conditions on growing domains. Yeah. So what is necessary? In order to get patterning, we have 4 conditions. Yeah, I have put here a key at the bottom. So I have 4 conditions. The first two conditions, they guarantee that my time-dependent US of t is asymptotically stable. This is no more than the trace and the determinant. The trace is negative and the determinant is positive. The third and fourth conditions are the ones that will drive this US of t to be unstable when I introduce diffusion. That's why you see the diffusion coefficients here, D, it's important. So what do these guarantee? Again, the last one ensures that I have real wave numbers. The K wave numbers are real. So what else should I say? I will say if we cannot, and this is also classical for Turing theory, in the absence of domain growth, if we remove domain growth, which are the red terms, remove the red terms, you have, for example, gamma f of u plus g of v is less than 0. You have minus gamma d times f of u plus g of v is less than 0. But if you multiply this by minus, I mean, there's a contradiction if d was 1. So if you plug in d to be 1, you immediately have a contradiction, yeah, between the first and the third one. Now let's assume that you introduce domain growth, and again you take d to be equal to 1. You will observe that you have 2 here, so 2 times h star, which is exactly this guy here. So if you multiply this with a negative, yeah, you get exactly this guy here in the third equation. But again, the signs are not changing, but this is a contradiction. If you multiply anything by a negative, when it's less than something, the sign should change. And therefore, d cannot be 1. In the absence of domain growth, in the presence of domain growth, d cannot be 1. So the activator and the inhibitor cannot diffuse at the same rates. This is only true, again, in the absence of domain growth, in the presence of domain growth. But when I introduce cross-diffusion, and this is important, I can relax this assumption. And therefore, cross-diffusion allows me to consider equal diffusion coefficients. Again, I'll overcome one of the major criticisms of Turing's theory. So that's it, really. I will skip this. I will concentrate on the differences because I think this is important. Just to illustrate why you might need an activator model. So if you go back, I'm just comparing the first condition and the third condition. If you compare them nicely, you can do the mathematics. It's not too difficult. You can show that the right-hand side is bounded by 0 from above, yeah? And you can write them in particular as d minus 1 h of star minus gamma times f of u. So if you assume that u is the activator, i.e. the derivative, the partial derivative of f with respect to u is positive, and you assume that g is the inhibitor, so g with respect to v is less than 0. In the absence of, and this is important, in the absence of domain growth, h star is equal to 0, you can show that you have this condition now, which is 0 is bigger than 1 minus d f of u. And this means because f of u is positive, you really need d to be bigger than 1 for this condition to hold. And that therefore it gives rise to this classical, long range activation, short range inhibition. Yeah. Yeah. So no. Yes. So long range activation, it should be the other way around here. So we need an activator inhibitor, but with this long range activation, short range inhibition. So if I now take h star to be different, then I can bound. It's not that I have just bound this quantity by d, but there's an h that I can play with. There's an h star that I can sort of play, and it has to bind to act as a bound, upper bound, for gamma times f of u. Yeah, you can divide by gamma if you want, and the f of u should be bounded by h star over gamma, for example. And if you can cook up kinetic side that is true, then you can have this idea of long range inhibition, short range activation. Yeah, so similarly, you can come up, if you rearrange the same inequalities I gave you, you can also show that G, which is the inhibitor, the inhibitor here, the GV is bounded by 2 h star over gamma. Again, it's clear. When h star is 0 in the absence of domain growth, G of V should be an inhibitor. It clearly. When h star is 0, V should be an inhibitor. But when h star is positive, you only need G of U to be bounded by the rate at which the domain is changing. And so there is no need for V to be really an inhibitor. It could be an activator, and that's what I'm proposing here. You can have an activator mechanism that will give rise to pattern. In the interest of time, I'll just show you some examples of what I was talking about. The growth rate phi here, you have linear, exponential, logistic. You can compute the h of t, you can compute the q of t, and I have some open problems here in understanding the long-term behavior of solutions as t goes to infinity, because as t goes to infinity for the logistic, for example, phi of t converges to k or kappa, and kappa minus kappa will be 0, h of t will tend to 0. So in the limiting case, you have these properties that your divergence is turning to 0. While for the exponential, your divergence never tends to 0. It's always constant. M times R is the growth rate. For the time dependent in terms of the linear growth, you can also show that h of t will tend to 0 as t goes to infinity. It's quite clear here because M times R is constant. So you have these properties, but there is quite a lot to be done in understanding what does this mean. Yeah. Some examples here are Turing spaces. These are domain-induced parameter spaces. The only one that's on stationary domains is the red one. Yeah, that's the only parameter space that comes out of in the absence of domain growth. All these other patterns, all these spaces, they emerge due to domain growth for an exponential. I wanted to illustrate you some of the time-dependent manifolds, but I will skip these. You can find them. For the linear and logistic growth functions, you have these ones as well. Again, the red one is this classical Turing space in the absence of domain growth. And when you introduce domain growth, you have spaces that are much richer, and you can widen your parameter space really due to domain growth. And that's the take-home message. I will just give you some solutions on an activator model. I say it here, which violates the cross and pure kinetic conditions, as I stated earlier. Yeah. So your diagonal entries are positive. So f of u is positive. G of v is positive. So v is an activator. U is an activator, basically. If you were to solve this in the absence of domain growth, you will never get patterns. I mean, you can try, you will never get patterns, period. That's the end of it. You introduce domain growth, either linear, exponential, or logistic, as shown in these three frames. So the first row is exponential, the second is linear, and the third one is logistic. You can see that the patterns are starting to form much later in time. Initially, there are no patterns at the beginning because the model does not give rise to pattern. It's only when a domain is grown sufficiently large that you start to get patterning. I am pretty sure that if I probably solve this starting from, depending whether I can cook up another reaction, kinetics for inhibitor, I need the domain to contract in order to be able to get patterns. For the cross-diffusion, I think I have time. For the cross-diffusion now, I introduce cross-diffusion here. It's a linear cross-diffusion with these terms. Again, it's non-dimensionalized. The rest of the model is exactly as before. I have dv and du. These are non-dimensionalized cross-diffusion rates or diffusion coefficients. Yeah. So that's the only difference. In terms of the theorem is exactly the same, but now I introduce in blue those terms that are emerge or arise due to cross-diffusion. Yeah. Now, it's important, I said before, that d cannot be equal to 1, but now I can set d equal to 1, and instead of just controlling the leading terms h star 1 plus d minus gamma d times f of u plus gv, I have these extra terms from the cross-diffusion that allow me to control the diffusion, the master diffusion d. And in particular, for the well positive of my PDE, I need the determinant of the diffusion metric to be positive. And this allows me to choose d equal to 1, but provided that the absolute values of the cross-diffusion coefficients are bounded by 1. For the well positiveness again. So this is very good. It relaxes, and as I said, it relaxes the Turing theory, as for pattern formation. And I will show you some examples where d is equal to 1. You can get, in fact, in some of my papers, I illustrate both the spaces as well as these patterns forming when d is equal to 1, as well as when d is less than 1. Yeah. So I will skip and go to some illustrations. This is exponential growth. Again, the way you read this is on a stationary domain. The first figure. This is when because the growth rate r is equal to 0 here. When I have a growth rate r is equal to 0.02, so I'm changing the exponential growth rate here. I'm taking d to be much larger than 1. So it satisfies the classical conditions, again, of activator inhibitor models or long range, short range activation. And so the red encodes only for this phase graph, only the Turing space on stationary domains. The other spaces are due to domain growth, yeah, and also cross diffusion. There's no domain growth here, it's only cross diffusion in the first one. Now the second one is coupled domain growth plus cross diffusion. And I'm changing the diffusion rates, the cross diffusion rates here, to illustrate to exhibit different patterns, parameter spaces really. In our papers, we have chosen parameter values from some of these spaces outside of the standard red space, and we've obtained solutions, stationary solutions. These patterns that I'm showing now, I'm choosing D to be equal to 1 here. So this is for the diagonal this is the diffusion of the diagonal entries. So they're equal. So the DU and the DV are equal. If I solve this in the absence of cross diffusion, even in the presence of domain growth, these parameter spaces do not exist. So I reiterate again, I solve the models in the absence of domain growth. No, in the absence of cross diffusion. Even if I have domain growth, I do not have these patterns, yeah, or on stationary domains. So these are cross diffusion. Maybe I should have put it here. Cross diffusion driven instability parameter spaces. Yeah. Again, very interesting. Similarly, for here, I'm showing here what I call short range activation, long range inhibition type of parameter spaces. D is very small, it's 0.05, and the other cross diffusion coefficients are given here. Just to demonstrate that this for linear and logistic, I can get patterns, and again for linear and logistic, look, I just pick parameter values from these spaces. These spaces are only induced by cross diffusion. If I take out cross diffusion, they don't exist. You know, even in the presence of what do you call it, domain growth. So it's important, really. What else? I was going to comment here that if you look at the initial frame, there's no patterning. Patterning only starts to emerge later on as the domain grows. And so I had both for linear and logistic. And I think I am on time. Yes, just on time. I would like to summarize. So Turing spaces generally change, and this is important, in size and shift location within parameter space once you take into account domain growth and cross diffusion. Yeah, domain growth implies that, you know, we no longer really need this standard paradigm of short range activation, long range inhibition. Yeah. This is a typo, really. This should be short, long range activation, short range inhibition. Sorry for the part type. I should correct this. And therefore, we can have what I call this should be again long range inhibition and short range activation. The other way around. Yeah, I apologize for the mistake here. In case yeah, it's just the other way around. So. What else I can have, and this is important, we can come up with activator, inhibitor models that will give rise to patterning. What else? In terms of open problems, I think one of my wish would be for experimentalists to have experiments where they can generate patterning, in particular in cellular biology, or plant biology. I don't know where your domain is changing in time. If they can have experiments carried out on environments which are time dependent, that would be very helpful. Then probably we can verify some of these. These are purely theoretical outcomes that have not been validated experimentally, and there is that need, I think. Also the need to formulate non-standard reaction kinetics, but we can only do this if we have some sort of guidance from experiments. I give some examples here of what type of work might be in this case in terms of formulating bulk surface PDs, domain-dependent analysis of these models. It's very important, and there's work that I'm doing with Wakil and Gusme and so on, Raquel. Also, Kusedu, we have a paper that we just are about to submit on bulk surface, which shows the wave pinning on a stationary domain. But the question is, you know, the dynamics are really revealing when you look at the long time behavior of your model. If you look at the short time scales, you don't see, well, you get the wave, but it gets pinned and you think, oh yeah, it's pinned, it's stationary. But if you were to solve for longer time, you see that, oh, actually after some time, the wave starts to move again. It's, you know, understanding that it's an important issue. Also understanding how wave pinning forms on a growing domain is an open problem as well. Yeah. The other problem that's important is to develop these numerical methods for bulk surface PDs, where we couple with mechanics. Continuum mechanics are important, in particular for cell migration, where cells are migrating through these complex environments, as I indicated at the very beginning. So I want to thank my funding bodies who have supported me over the years, and they are reflected here just for their benefit, really. So I will stop here and probably stop sharing. And then if you have any questions, you can ask.